If I can have the first slide. This is my thank you slide and my conflict of interest slide. So these are some of the groups that have helped us bring this from an idea into reality that everyone can get access to this. And what you really want to know is what's next. What's next is coming much faster than it's ever come in any other field of technology that I'm aware of. It's amazingly rapidly approaching. We may see sequencing or some sort of molecular biology that is part of your phone at some point. That could be a ways off, I don't know when. We may be able to see this inside of your tissues at a high granularity. This is called in situ. We are certainly already seeing interpretation integration software that includes genomics, and this is incredibly important. And most important, one of the things I'm going to emphasize with the technology is that we need really great cohorts of people like you, who will help us to interpret the genome by sharing something about their own medical and life experience. And this cohort will have the advantages and the disadvantages of being the first people to test drive this personalized medicine, just like the first people to drive a car. Now I'm going to emphasize the importance of extremes. Everybody in this room may think that they're normal, that they're in the middle of a bell curve, but in terms of medical progress, there's a lot to be said by looking at extreme cases. Here's an extreme case of this young man named Flex Wheeler, who had the unfortunate situation of being deleted for both copies or mutated for both copies of his myostatin gene. As a consequence, he has decreased body fat and decreased chance of atherosclerosis. It's a pity, isn't it? Anyway, he's in every other way completely normal, and exactly the same genetics and the same phenotype is reflected in these three different animal models: mouse, cow, and dog. So it's very likely this is the cause of his increased muscle growth and low atherosclerosis. So when we look at these bell curves or other distributions, we're really interested in the extremes. And the reason is that the prevention and cures we're looking for can actually be very creatively far from the discovered cause. You have a minor allele, and we'll see this in a little bit later on, can be the cure for the major allele. The major allele isn't always the best allele. Gene therapy doesn't mean just fixing a minor allele. It can be used to introduce something that's brand new, like a reaction to a particular cancer. And I've already mentioned the extremes, and these extremes include looking at people who live a very long time, as Ernst Hoffmann has already mentioned. So when we're talking about interpreting genomes, we're not talking about just going from your personal genome, which might have 3 million differences between you and me, and then going and telling you exactly everything about how you're going to be, in spite of the fact that identical twins are so similar. We don't need to do that because we have lots of other data, and all of these, which include how your genome interacts with your environment, these are heavily intertwined. It doesn't make any sense to say that it's one or another. Furthermore, we have the opportunity to measure many other things with these molecular tools. So these molecular tools have come down in price by about 1 million-fold in the last few years, and it's affected not just our ability to measure our personal genome, but many other genomes, such as the microorganisms that exist in your blood, or your skin, or various other tissues, and your immune response to them, and allergens, and so on, and how this plays out in your body through what's sometimes called epigenomics. And you can monitor these from an increasing number of tissues by doing stem cell reprogramming. Now, I'm a little obsessed about reducing costs, making things accessible to everyone, to lower the price. And the interesting thing is this isn't just lowering the cost for developed industrialized nations. This is lowering the cost worldwide. And what you find is you might bring the cost down for cataract surgery to $3,500 in the United States. But then, and one of my good friends, Jeff Tabin, has repaid his debt to the Sherpas in Nepal by teaching them in outpatient clinics how to bring this cost down to $20 locally. Now, why is it that we can even consider bringing down healthcare costs? Because everybody talks about healthcare costs skyrocketing and so on. Here's an example where the costs have not only been improving exponentially, these are factors of 10 on this y-axis, just like the electronics industry, but that exponential just got even steeper in 2005, so-called next-generation sequencing and synthesis. This means that we're going now exponentially 6 times faster than the computer industry, which is already going quite fast. Why are we going so fast? I mean, what allows us to go that fast? And they're now not one, for most of the Genome Project, there was basically one technology. There are now 37 next-generation technologies, of which 9 of them in the red box here are already commercially available. And then many others that have demonstrations of various sorts. And you're going to see, I think, in the next couple of years, this green box, which are nanopore technologies, coming into being. Typically what these ones that are already available have in common is the ability to do multiple probings of a flat piece of glass or other container that's flat, in which you can image over and over, looking at billions, up to billions of points of light at a time. Now we'll come back to technology in a moment, but I want to interject that technologies are only as good as how you share them these days, especially. So if you're the only person in the world who has a cell phone, that's not very useful, even if you're very wealthy like this guy. So you need to develop ways of sharing, and this is particularly true for genomics, because you not only have to share the network, you have to share the information, and you need, in order to get interpretation of that information, you need to have traits, environments, and genomes for many people, and you can't just do it in model organisms, as Ernst said very articulately earlier today. So we're talking about open access cohorts to test drive personalized medicine, and what's happened is that each of these technologies has come online. They've wanted to basically show off how well the technology works by sequencing a human being, typically one who is identified and is willing, in many cases, to share their personal data. In particular, Personal Genome Project, which I'll tell you a bit more about in a couple of slides, Personal Genome Project has been greatly aided by the Illumina and Complete Genomics platforms, where many of these individuals have been done for free. Now, what is the Personal Genome Project? It is a worldwide organization that now has 16,000 volunteers in 81 countries. This is a little out of date. And what it aims to do is make genomes, trait data, and cells available without strings attached, without any complications for academics or companies that want to use it. They don't have to worry about whether the individuals have been properly consented and whether the data can be shared or has to be locked up, because all of these individuals have been consented with full sharing. And the reason that we are sure of that is that we now have 2,400 of them that have achieved not a passing grade on an entrance exam, but 100%. We require that they be 100% knowledgeable of what they're getting into. We don't hand them a consent form, 10 pages, and have them sign it knowing that they haven't read it. That's very commonly the case for medical genetics. Here we require that they show that they know what they're getting into. We have 500 complete genomes in the pipeline right now, in addition to the ones I showed on the previous slide. There is a revolution going on, and the distance between research and application in a clinical setting is no longer decades, it's weeks. And here's an example that actually, the covering of this study by journalists actually got them a Pulitzer Prize this year. This Nick Volker was 3 years old, and he had dozens of surgery on his intestines, and it was very clear to his pediatricians that this was not going well. So his pediatrician said, was far-thinking and said, we should sequence this person. And he looked around until he could find somebody who would sequence Nick Volker, and on the basis of that sequencing, they immediately changed their opinion as to what was wrong with the child and changed it from a focus on intestinal surgery to a focus on a hematopoietic stem cell transplant. He was cured by this in a few weeks. The causative gene is here, XIAP, and he's in fine shape ever since. So that's weeks, not decades. And that's not just an isolated anecdote. You may get the impression from some of the people that are concerned with direct-to-consumer genetic tests that genetics is a toy or is not working well. I would strongly disagree. Genetics is very mature. It's been around, human genetics has been around since 1963. There are now 2,400 or more diseases, including even more genes that are considered highly predictive and medically actionable. These are not toys. These actually work quite well. And BRCA1 and 2 and hypertrophic cardiomyopathy examples. Furthermore, we can not only sequence the genome as you're born with it from your mother and your father, but we can see how it plays out in your various tissues. Blood, saliva, skin, these are easily accessible, but we're going to more and more challenging tissues, in part by doing stem cells, in part by doing less invasive biopsies. But here we can show that we can sequence on these cells, these are actually cells, where each of these dots is a different messenger RNA molecule, and we can very inexpensively see as little as a single base difference between these two cell types here. The green and the red only differ by one base pair and one messenger RNA, and we can do as many messenger RNAs as we want. Here's an example where the difference between the cells is some of the cells are fine, and some of them have cancer, and some of those cancers are drug resistant. And you can monitor these little green dots that go from almost no green dots to lots of them, means you're building up drug-resistant cancer, and it's time to switch the drug. And you want to catch this as early as possible. It's like looking for a needle in a haystack. It's finding a very rare green cancer cell here in this case. And again, this can be as little as a single base pair that changes one position in one protein that causes resistance to these anti-cancer drugs. Speaking of, that was leukemia, and this is also a story about leukemia, but it's a very interesting, what I consider a tipping point, turning point in the way we're thinking about genetics and, in fact, normal medicine. This individual had both leukemia and AIDS. He had AML and HIV infection. The typical treatment for AML was to find someone who was matched with him in terms of being a donor match, meaning having his transplantation antigens which were compatible. They managed to find someone who could act as a transplantation donor for hematopoietic stem cells, who was not only matched, but also happened, this individual was donating the blood precursors, happened to be deficient in both copies of their CCR5 gene. That meant this person was a very rare person. And I'll tell you exactly why that's important in a moment. So they transplanted these blood precursors from one person to this patient, and he became cured of AML and HIV because the blood now that developed in his body from this donor was resistant to HIV because the HIV receptor is CCR5. This was a serendipitous occurrence in the population, but now it's being commercially developed so that you can make a CCR5 deletion whenever you want in your own cells And these are now in phase 1 clinical trials, and the results so far are very, very encouraging. You're making a precise change, so this sidesteps many of the problems with the earlier gene therapies. And the important thing here is not just that gene therapy is beginning to work again after a decade, but that this is, you're taking someone who has the common allele and replacing it with someone who has a rare allele. That is to say, the rare allele, like that myostatin example I gave at the beginning, is in a certain sense better. They're resistant to HIV. Now, as you do these stem cell transplants, you want to make sure they're high quality. You want to make sure that what you're delivering to the patient in terms of stem cells is the best it can be. And it turns out now, because sequencing is so inexpensive, you can use this as a quality measure to make sure the quality of your stem cells is high. And we've been looking at some very, very recent protocols for making these so-called human-induced pluripotent stem cells. And we have found there are errors in every single one of them so far that we've looked at. And they affect coding regions, and many of these coding regions are in genes involved in cancer. So it's very important to do this kind of quality control. In the Personal Genome Project, we are establishing stem cells for all the participants, and this constitutes a biobank, which are then trans-differentiated into every major type of cell, just as jargon for endoderm, mesoderm, and ectoderm, derived cells like respiratory epithelium and so on, and also liver cells. And we can now study these with pathology tools, with proteomics, and with RNA. And we can find inter-individual variation here, not just in the protein level, but also in their activity. Now, the beautiful thing about the Personal Genome Project is not just that we have traits, environments, and genes for each individual, but as time goes by, they become more and more valuable as a cohort, in that we've done so many things on them that if you want to see the confounding effects of one environment or one gene on another, you have already collected that data. And so, for example, when someone wants to do a new study on functional magnetic resonance imaging, brain imaging, this is actually my brain. I'm PGP number 1. This is the previous slide where it was PGP number 1 and 9 were compared for their trans-differentiated tissue types. And here's PGP number 2, which is John Halamka, who's had a RFID. These are the sort of things you use in the store to identify merchandise, injected into his arm. And this is the beginning of quantitative monitoring, real time, all the time, without of your body physiology. So we have testmybrain.org is cognitive and facial recognition, and then we have this MRI, looking for variations from person to person. It's not just, these traits can include traits that are also environmental components. How your immune response to microorganisms is a relationship between you and your environment. And when you're doing this as a diagnostic test, you have a choice between testing for the microbes themselves, which is very important if you're trying to catch it fast, or if there's a drug resistance, because very often the immune response doesn't reflect drug resistance. But the immune response has the advantage that if the pathogen comes in, causes some damage, and then leaves, you can catch the immune response to that pathogen. So this is the method of choice, for example, for HIV and tuberculosis, is looking at the immune response. So we wanted to generalize both of these. How can we detect all the microorganisms at once? Usually it's done one test at a time. And how can we measure the entire immune response? So for the immune response, we actually look at the entire B and T cell lineages as a function of time by taking blood samples. And this is reading out not only what it sees of microorganisms and allergens that might be present in your body, since the blood goes to every part of your body, but it's also seeing antigens that might be reaction to your self-antigens, if you have a cancer that's developing, or your autoimmune disease. All of this is reflected in your immune response, and we can monitor that with time. So here's an example of a Personal Genome Project vaccination time series. We've now been monitoring over a 3-year time series. This is 3 of the PGP. Again, this is me here. and these are two younger, 20-year younger people. And what you can see is relative to the vaccination, this is the seasonal flu virus vaccination, at time 0, at 7 days out, you get this huge spike of these healthier young people of particular immune responses that turn out to be aimed at mostly at the virus that we're using for the vaccination. And we can track these over time, no matter how they change, whether they can change their constant regions or their position in the body and so forth. We can track them because it's exquisitely detailed barcode that kind of comes from the recombination and mutation that occurs naturally in the immune system. So we can follow the immune system in its response not just to pathogens, but to vaccination, allergens, and cancer in your own body. Furthermore, we're beginning to, as in this Nature Biotechnology paper that we did in collaboration with Stephen Elledge, we're beginning to come up with ways that we can identify the relationship between a particular antibody and the antigen that it reacts with by making libraries of antibodies from human blood as a function of time, and making a constant antigen library, which is in this case all the human proteins, all the segmented by tiling every 36 amino acids throughout the entire proteome. Every human protein is put into this phage library expressed on the surface of viruses, and then we can bind the antibodies to the viruses and find which antibody goes with which virus. So I've given you an overview of where I think not just the technology is going, but our ability to apply this and interpret it. I think it's key, if we're going to interpret it, to do this in the context of the therapies that we're trying out, whether they're the right therapies or not. This is part of our environment, how our immune system interacts with microbiomes, toxicity measurements, which we can get from our hair and our nails, the nutritional monitoring. All of this, we have to get better at monitoring our environment. And this entire test bed, which is very experimental now for the Personal Genome Project, will be considered routine in a few years. And it'll be very interesting to see which parts of these drop in price and which ones turn out to be not quite as useful. I suspect they're all going to be quite useful. We just have to continue to work at integrating them and developing software that makes it very easy for decisions to be made. Now, the Personal Genome Project was developed in the context of an ethical project. It basically was to ask whether we could do better than the current methods for consenting individuals, where you made promises of privacy which were hard to keep. It has grown from that ethical standpoint, and we continue to go back and reassess all sorts of aspects of this, and I'd be delighted to talk about them. We keep going back to that and trying to improve the procedure by which we consent individuals and educate them to make sure that they know what they're getting into, and ultimately return to them value to them, their families, and other people around the world that have the same genetic and environmental components that they have. So, period, full stop. I hope I've stimulated some conversation here. Thank you.
Thank you very much, Dr. Church, for this exciting contribution. May I perhaps...
Can we take questions now?
Sure, sure. May I perhaps...
There's one right there.
Yeah. Thank you very much for this presentation. I think this is sort of drives things much more forward than where people are right now. And I think this is, we should be thinking very much along these lines. The one question that I have, and I still in my mind have tried to resolve and not sure how to yet, is if we're trying to capture environment, we can sample a whole bunch of things that we can sample of a given individual, maybe over a little bit of time that we have access to this individual. But it's kind of harder to predict or to infer what has happened in the past. And one idea that is emerging now from at least a few people is that we can actually sample the current cellular environment, but not through IPS because that's reprogrammed, but through tissues to get some epigenetic marks that are representative and consequence of the variable environment. The problem with that is that which tissues and how deep can one go to sample enough to make it worth? Do you have sort of an idea of how we can go about doing this, if you agree that this is a reasonable way to do it?
I think it is very reasonable, and there is a tendency to try to do it with what's easy, and I don't think that's necessarily the right approach. I think the best approach is to bring down the cost of what's the correct thing, even if it's hard. Just work. So, for example, I think one of the things we're excited about is a generalization of the idea of circulating tumor cells, sometimes called CTCs. That gives you access to cells in a variety of different tissues. They tend to be pathological because they've breached the endothelial layer and so forth, but there's a certain amount of reflection of many tissues in the blood. So that's an act of convenience, however, but it is one approach. The other is there's increasingly better ways of getting biopsies via catheters. So, for example, my cardiovascular colleagues think nothing of taking a heart biopsy. They consider it non-invasive, which I think is very interesting. So that's another approach. And then the final one was the one I mentioned with the immune system. The immune system goes around and does surveillance where the immune cells actually go through the endothelial wall. They even breach the blood-brain barrier. And then some of them circulate, and you can get access to those. We need ways, we are developing ways to pull out those cells so that we know which is which, or just sequence them all, and then we know by computation which one is which. But it's a very good question. You need access to all the tissues, not just the ones that are convenient.
May I perhaps just follow up a little bit
sure.
on that? Is you also said that when you sequenced stem cells, you detect a lot of errors.
Yes.
What are you going to do with those? Are you using them, or?
So we have just gotten to the point where it's inexpensive enough that we can do multiple sequences from the different body parts of the same individual, including stem cells. We think that this particular observation is due to a crisis that occurs in reprogramming from skin to an embryonic-like state in the induced pluripotent stem cells. That means there's an opportunity for improving the protocol, which we think has something to do with oxygen stress and probably some other things. So there's protocol improvement. We're also testing the hypothesis. We know that we can engineer these, and during the engineering, ironically, you get fewer mutations during homologous recombination engineering than you get during establishment of the stem cells. So in principle, we could fix the errors. These are two different avenues. One is protocol improvement, the other is fixing the errors and then redoing the quality assessment genome-wide to see that the fixed version of stem cells is better. I don't know which is going to work out better, but I think the best approach is to try every technology you can think of and make it cheaper.
Okay. Ernst?
George, you mentioned in your consent form, or actually the test that your participants in the Personal Genome Project have to do, you have to be 100% successful in doing this test. And I think this is very important for this sort of quasi-experimental group. But this is certainly not something that scales because, I mean, you will not be able to educate everyone to this level to profit from it. And in the past, people went to the doctor because they wanted the doctor to tell them what's wrong with them. And so they didn't study medicine to make some changes. So how are you going to scale this?
Well, actually, we think this is scalable for the following. This is the original protocol that our institutional review board required was that they have a master's degree in genetics. This test is by no means showing that they're expert in genetics. This shows that they're experts in the risks and the benefits, if any, of participating in the study. It's basically walking them through the consent form and some very basic genetics so they can explain to their family what their family is getting into. Because unlike many, so the ethics in many countries, including the United States, is if you sign up for a project, you do not have to consult with your family. It's the individual that makes the decision. But in our project, we actually want them to consult with their family. In fact, we want them to bring their family with them if possible, but at a minimum get their, so that's what the test is about. And the reason I think that everybody can pass this test, well, first of all, many of the people that have gotten 100% on this exam are absolutely ordinary people, no offense. They're average, they're average education. And they're allowed to take the test as many times as they want. So in a way, the test itself is an education. They don't have to be pre-educated before they get in. But it's a great question, and we inevitably there will be people that are excluded from all medical research. There are people who simply don't want to participate in medical research, but we're trying to make it as easy as possible.
One more question here.
Thank you very much for your talk. It was very interesting. I was just wondering your view on gene therapy, because you showed us this really nice example of bone marrow transplant that could heal AIDS. So I was wondering, what is your vision of how far can gene therapy go in the future? I mean, can we heal heart muscles or something like that?
Right. So I think we're seeing a renaissance in gene therapy. It's not, and it hasn't been heavily commented on in the press, I think because there's this perception that it failed tens of years ago. Anything that failed that long ago is never coming back. But not only can we do this, make virus-resistant cells, but we also, there's a beautiful example that just got announced where you do gene therapy on T cells, where you put a specific receptor on its surface that will kill off a B cell leukemia, and that has caused remission over a 1-year period. So there may be now, this is one of the first successes in an anti-cancer gene therapy by changing engineering the T cell. And then finally, your question about cells that maybe are not floating around in the blood is a harder case. And there's been progress on doing retinal cells and other cells of neuronal lineage, which is in a way the worst case scenario because they're not only not migrating, they're not dividing. But this has also been successful, including one of my postdocs who did during his thesis did putting in a channel rhodopsin gene into retinal cells so that they now can respond to light when they couldn't before.
May I also have another completely different question, perhaps? Now you show us that the costs for sequencing and getting access to genome sequencing are becoming lower and lower and lower. But is it not so that the phenotyping, the costs for phenotyping, that you can actually then start to correlate in a meaningful way? You are probably developing in the opposite manner. When you want to phenotype them completely, that this information is most useful.
Yeah, so the question, just to restate it, is if the genotype goes to 0, is the phenotype going up, going down, or staying the same? And I think there are a few things in medicine that are going up in price, and I think that's telling us that those are the wrong strategies. But I don't think that the phenotype is actually going up in price. I think it's coming down, partly because electronics is getting less expensive. And so the ability to monitor yourself with, say, something like a cell phone is becoming increasingly likely to occur. In other words, the cost curve for electronics will make it possible for you to measure not just blood pressure and heart rate as an athlete might, but many biochemical assays as well. That's not what we have right now, but I don't think that's going to be expensive. I think that's actually going to be quite inexpensive, and everybody who wants to will be able to do it. Okay. So that's one kind of phenotype. Another way that phenotypes are getting cheaper is that medical records are now becoming increasingly electronic. And so, to some extent, all of your phenotypic information should be in your medical records. And as healthcare providers become more and more electronic, then in order for a researcher to harvest that information is simply a matter of making an agreement with the patient, which in the Personal Genome Project, they download their entire medical records. And that costs us nothing.
Okay, thank you. More questions? Yeah.
You mentioned the precision of the sequencing of RNA. And in the nervous system, at the time there is a boom of the expression of messenger RNA in function of neuronal activity. My question is, what is your expectation when it will be possible to make local sequencing of RNAs in structures affected, for example, by identification from the imaging situation?
Let me make sure.
You describe the imaging situation, and then the question is precision of the sequencing of the messenger RNA. Is it feasible now to say we will go in this type of brain structure and have let's say, a pedigree or a profile of the sequencing of messenger RNA affected by this type of neuronal reaction, let's put it this way.
Right, so the question is, these methods should be applicable, they're currently applicable to a variety of cell types, including neurons. And they're high resolution in the sense that you can see subcellular resolution, so you can actually see the difference, if there is a difference in distribution of an RNA, say between the axons and the cell body of it, and you can see the impact of environment, whether that's adding a drug or adding a behavioral environmental component. We haven't done that much in the nervous system, but we have a collaboration with the Allen Brain Institute where they mapped all the RNAs in multiple brain sections in multiple brains. We'd like to redo that where we do it all in one brain, because now we can measure multiple RNAs sequentially in the same section, because we're no longer limited to one color or 4 colors. We can do hundreds of colors effectively by reusing the colors over and over, as you do in next-generation sequencing. This is basically next-generation sequencing combined with in situ hybridization, to give you the power of both.
Okay, thank you once more, Dr. Church. Now,